fibrosis is a genetic disorder, and this disorder is due to a mutation in the gene responsible for the formation of the CFTR. This is a transmembrane protein found in this as a genetic disorder due to mutation in the gene responsible for the formation of this transmembrane protein. This membrane protein is found on the plasma membrane of many cells in our body, like the cells found in the respiratory tract, cells in the oviduct, cells in the sperm duct, cells in the pancreatic duct. This protein, when it's found in the plasma membrane, it helps in the pumping of the chloride ions out of the cells. And this is going to increase the water potential of the cells, so water will move out by osmosis down water potential gradient. This water will start to dissolve the mucus secreted by the goblet cells, and it makes it easier for the cilia to push away the mucus. Okay, so that's the role of this CFTR. This gene is found on the autosome, chromosome number 7. And there are two different types of mutations, and they both result in a recessive allele. One of the mutations will result in a different amino acids being found in the protein, and the cell machinery recognize two different types of mutations. في واحدة من mutations, it just changes one of the triplets, so only one amino acid in the protein will be changed. But still, the cell machinery recognizes the protein as something foreign, and it doesn't put it in the plasma membrane. So this protein will not be found in the plasma membrane. The other type of mutation introduces the stop codon. And so the polypeptide chain is not made and the protein is not made. In either cases, the CFTR is missing from the plasma membrane, and chloride ions will not be pumped out and the water will not be fused out. This will result in the formation of thick, dehydrated mucus. And this mucus, because it's too thick, it cannot be pushed away by the cilia, so it accumulates. People suffering from this condition have to be homozygous recessive, because it's a recessive allele. So people who are homozygous recessive will suffer from cystic fibrosis. The main cause of death of cystic fibrosis is due to recurrent infections of the lungs. Due to the accumulation of the mucus, bacteria accumulates and it leads to recurrent lung infections. Also, they have impaired digestion because the pancreatic duct is also blocked by the mucus, so the pancreatic enzymes do not reach the duodenum. Maize becomes dry because the sperm ducts are blocked by mucus, so the sperms do not reach the urethra. So it causes many problems, but the main cause of death is due to degeneration of the lungs. There is no specific treatment for the cystic fibrosis. The treatment is symptomatic. So when they get lung infections, they take antibiotics, they aspirate excess mucus, they take enzymes, oral enzymes to help in digestion, in the process of digestion, and so on. Recurrent infections. Coughing. Aspiration of the mucus. They remove it, they show the mucus is allowed. Okay. Anyway, we now have, due to the development of gene technology, something known as gene therapy. So gene therapy means that we are going to add the normal allele into the cells. And we have two lines of gene therapy. We have somatic gene therapy, and we have the germ-line gene therapy. If the disorder is due to a recessive allele, it will be easier to treat it using gene therapy. And just introducing the dominant normal allele is going to be expressed and the condition will be recovered. But if the condition is due to a dominant allele, it's very difficult to treat using gene therapy because you have to remove the dominant allele from the cell and add the two recessive alleles, so it's much more difficult. So it's easier to treat genetic disorders due to recessive alleles by gene therapy. In case of somatic gene therapy, they are introducing the normal dominant allele into somatic cells, that's the body cells. So for example, in case of cystic fibrosis, they are introducing the dominant allele into the lung cells. So they put this allele into a liposome, a lipid sphere, and they put it in aerosols, and the patients inhale this liposome, and it's going to carry the allele into the cell. So that's somatic line therapy. Of course, somatic line therapy have many drawbacks. Number one, it has to be repeated because of the short lifespan of the cells. The liposome is a lipid sphere surrounding the gene عشان يقدر يعدي من الفوسفوليبيد العائلية. بيحطوها ببخاخات aerosols بخاخات والبيشنت بيinhale فبيدخل through the airway passages وبيعدي من الممبرينز بتاعت الالفيولاي ويدخل السيلز. كده انت دخلت النورمال allele into the cells. المفروض بقى ان هو يمسك في الدي ان اي and starts to be expressed. انا لسه بقى بتكلم على الدروباكس بتاعت البروسيدور. One of the drawbacks in number one, you're targeting just one area. You're targeting only lung cells. لكن السيلز بتاعت البنكرياس is not targeted, بتاعت السبيرمز is not targeted, فانت targeting area واحدة. Number two, it has to be repeated always. لأن الcells have a short lifespan. Even when you introduce this allele into the cells, you're introducing them into few cells, some cells. But these cells will die, so you have to repeat it. The third drawback is when the gene is introduced into the cells, you don't know where it's going to be incorporated in the DNA. You don't know where it's going to be attached, in which part is going to be attached in the DNA. It could be attached anywhere in the DNA. It can switch off another important gene. In many cases, it led to cancer as well. Okay? So that's the problem of somatic line therapy. ما هو it's a gene, it's going to bind through the gene.
لا مش ممكن تدخل كده
لا it's a gene like all your genes. الدي ان اي نيوكليوتايدز كلها واحده.
لا قصدي ايه اللي هيخلي الدي ان اي يفتح ويدخلك؟
هتخش وقت الريبليكيشن بتاع الدي ان اي في اي حته.
بس كده عشان تخش القليل اللي هو
ماليش دعوه بس القليل الغلط ده ريسيسيف. القليل اللي انا دخلته ده دوميننت فالدوميننت هو اللي هيبقى اكسبريس. لا هيثم انت كنت عايز تسال حاجه؟ طيب ايوه يس. طبعا غلط طبعا غلط بس هي الايديا ان الناس اللي هم عندهم cystic fibrosis هم بيوصلوا لمرحله في حياتهم هم ميتين فهم they don't mind ان هم يبقوا experimental individuals for certain researches. They experiment on جايز وهكذا تنجح. فاهمة ازاي؟ It's a genetic disorder, mutation. منتشر جدا في اليو كي. منتشر جدا جدا في اليو كي. هو موجود الميوتيشن هناك والبريدنج الفريكونسي بتاعه بقى عالي. ايه؟ ملوش علاج، لا العلاج بتاعه كله سيمتوماتيك يعني السيمتوم اللي التاني عندوها. دلوقتي بدأوا بقى بالجين ثيرابي بس الجين ثيرابي زي ما انا بقول لك ثواني الجين ثيرابي زي ما انا بقول لك لها دروباكس كتير. في دلوقتي اكتشفوا دراج the patient can take this drug لان هو احنا قلنا ان one type of the mutation is a stop code being inserted in the middle. فلقوا drug that can allow the transcription to continue skipping the stop codon. It works somehow like that. فالبروتين هيبقى made at the end. And طبعا using this drug is better than the gene therapy. Number one, it doesn't need an experienced medical personnel. The patient just takes the drug at home. It has less drawbacks and side effects than the gene therapy. Okay? Yes.
دي هتبقى جنرال تارجت الانترست.
بس هم ليه ما يحطوش القليل ده في التارجت يعني؟
ما هو ده الجيرم ثيرابي اللي انا لسه هتكلم عليه. Okay? The germline therapy نعم يا عمر.
اللي هي بتبقى سكيب دي مش هو برضه فيها ماينس ناقص.
ايوه بس ستيل بتبقى better الكونديشن
يعني بتعمل تتحط
بتتحط بس it's not working as good بس بتبقى موجودة. طيب ال second line of gene therapy is germline therapy. So the germline therapy is when they introduce the normal allele in the gametes or in the zygote or in the embryo cells. Like for example introducing it into the oocyte. So all the cells will have this gene. Gene-line therapy is not authorized to be carried on humans. And we don't know what these babies are going to become. We don't know how they will look, what will happen to them. And again, you don't know where this gene is going to be attached. That it cannot be carried on, it's not authorized. It's illegal. It's illegal.
هو اللي بيحصل يعني؟
هما بيجيبوا الأوسايتس وthey introduce the normal allele into the oocytes قبل fertilization وبيعملوا in vitro fertilization. Now all the cells of the embryo and the cells of the fetus will have the normal gene. لكن it's not authorized, it's prohibited to be carried on babies.
ليه؟
On humans. لأن أنت ما تعرفش long term effect of this what would happen. أنت ما تعرفش الجين ده لما هيتحط هيعمل إيه؟ هيغير في إيه؟
مش انا هحاول احطه.
لا أنت ما بتحطوش، أنت بتدخله في السيل وخلاص هو بيمسك بمزاجه. ما هم ممكن يجربوا في أنيمالز لكن ممنوع يتجرب على بني آدمين. ما لسه الحاجات كلها تحت التجارب.
بس هم يعني مش هو الجين بيكون ان هو يخلي ده ما يحصلش. ليه؟
You don't know where it's going to be attached in the DNA. Well, that's the problem of all genetic engineering. انت ليه بتقول ان في hazards للgenetic engineering even in genetically manipulated crops or genetically manipulated microorganisms. لان انت you don't know what's going to happen to this organism that's being genetically transformed. It could turn, يعني مثلا انت sometimes when you use for example virus vectors or when you use certain kinds of bacteria to be transformed, they turn into aggressive pathogenic bacteria with no cures or aggressive pathogenic viruses. Changes happens, you're changing the genetic makeup. You don't know this change in genetic makeup هتعمل فيهم ايه. When you make genetically manipulated crops, why is it now a hazard to eat these genetically manipulated crops? Why is the whole world against GM crops now?
كانسر.
لأن long term effects بدأت تبان دلوقتي.
ايه بقى؟
الكانسر. اللونج تيرم افكتس بدات تبان دلوقتي. هما بقى اول ما طلعوها وقالوا ده بقى احسن حاجه للهيومانيتي، هما they didn't know the long term effects اللي هتظهر بعد كذا سنه. Okay? Plus ان مثلا a lot of GM crops turned into very aggressive weeds and they went out of control. They grow very quickly and they destroy the ecosystem اللي موجوده في المنطقه. وبعدين المشكلة برضه GM crops في ايه؟ GM crops is going to contaminate organic crops لأن once يعني it's better to use genetic manipulation على microorganisms أو unicellular organisms than you're carrying it in labs, in fermenters, in closed areas. if a problem arises, you can just destroy the whole batch. Like if you are carrying out a genetic manipulation on an organism, you're introducing it directly into the environment. You are carrying it on a crop, on a certain plant, or on a certain human, or whatever. The gene is now in the environment. You cannot get rid of it. But if it makes any problems, if it makes any hazards, it's there. You cannot get rid of it. خلاص بقى موجود في الانفايرمنت.
طب هو وين؟
سمعت ليه ما يستخدموش سبيشال انزيم زي
لا ما وصلوش لده.
ما
ما وصلوهاش.
هي اسمها جيرم لاين
ايه؟ germ line لانها it was carried out in the gametes. هو ده الجميت لاين. هو اسمه كده.
هو كده تمام اهو.
ما هما الساينتست بيطلع بايديا and he wants to try it. فهو he has to make هو بيطلع ببحث وريسيرش وبيقول فكرته وبعد كده بيبدا يقدمها وبيتوافق ان هي تتعمل ولا لا. فهو up till now الريسيرشز دي كلها ممنوعه على الهيومنز. لا دي حاجه ثانيه. لا دي حاجه يعني هو دلوقتي في حاجه اسمها ده اللي احنا هنكمله دلوقتي.